It's a delight to be back here at Mesa College. I was here in 2007. I hadn't forgotten. I looked at my old notes and slides, and they're way out of date. So it's apropos tonight to come here and tell you some updates in the research area that we do. But before we do that, if you would indulge me, I know it's totally passé, but I'd like to get a selfie. Is that okay? All right. It's getting hard here. It's a big group. All right. Thank you. The institute that I work at is a nonprofit research institute. It started 40 years ago as the La Jolla Cancer Research Foundation. And its focus for quite a few years was, in fact, on cancer. But over the last 20 years, it's grown into an institute that focuses on all major health-related issues. And we're particularly focused on understanding the fundamental processes of physiology in the immune system, in the central nervous system, and how cells go awry and become malignant or become infected with infectious agents, and translating that information into effective therapeutics or treatments that will alleviate really some of the scourges of mankind. That's our fundamental principle. So what I'd like to do is to start off a little bit and challenge you. You are all aware of our 5 major senses, and the one I want to tell you about tonight is your 6th sense. And of course, it's not Haley Joel. He was a little bit out there in that sense. However, our 6th sense was absolutely critical for winning The War of the Worlds, an H.G. Wells novel. And as you may recall, this film and its importance and why these creatures died because they didn't have a 6th sense. Your 6th sense is your immune system. It's your ability to survive in an environment, but it also contributes not only to the external environment, but it peruses your internal genomic makeup in a way that's very unique and very effective in keeping us alive in a very hostile world. This picture here shows you an electron micrograph of one of the principal players in the immune system. It's a lymphocyte. And I'll tell you a little bit about these lymphocytes and what we know, but I thought I'd give a little bit more of a general introduction into what the immune system and how it functions as a defense mechanism against infectious diseases, but also how infectious diseases teach us about how the immune system works. And I'll tell you an example of that as we move forward. So, host defense is a general term for protecting you against environmental insults. And these environmental insults are typically infectious agents. They're part of the microbiome or the virome, viruses, fungi, and bacteria are the elements that we face continually in our lives that want to eat us and use us as a food source. And your immune system allows us to exist in that environment. If you don't have an immune system, you can't live in our environment. The boy in the bubble is a classic example of a young man who had what's called severe combined immunodeficiency. He had neither T or B cells and was incapable of responding to environmental pathogens. And so he had to be kept in a sterile environment. So when we talk about inflammation and host defense, the inflammatory part of the immune response is, one, you have to identify, you have to recognize that there's something there. The immune system recognizes molecular shapes. And those molecular shapes are often found in the shapes encoded for the molecules like proteins and nucleic acids, and combinations of those in the presence of carbohydrate, macromolecules, if you will. It's what our genome encodes and what produces and allows us to be functional elements. So once a foreign substance is recognized, or an antigen as we call it as immunologists, we call foreign things antigens, it has to isolate, And by way of isolating it, it changes blood flow or lymphatic flow. And that helps contain that organism at a specific site. The immune system is then equipped with multiple mechanisms of destroying that tissue or cells that are infected, or destroying the microorganism like a bacteria. It has multiple mechanisms of doing that. This is an incredibly powerful system because it literally can reject any cell in your body. And in fact, when we do an organ transplant, you all hear about transplanting a liver or transplanting stem cells from some other individual. Our immune system is capable of recognizing differences between human beings. Those are allogeneic differences, as opposed to differences in species or in microorganisms. Those would be xenogenic differences. The genetic background are very different. But within the human species, we have lots of different genetic variation in our genomes, and our immune systems can detect those differences. And in fact, they're selected during development to detect those differences. So the immune system has this incredible power to destroy other cells, and in fact, it'll do it when you don't want it to. And there's a whole host of diseases known as autoimmune disease where your immune system attacks its own body and ends up destroying those cells in the process and leading to a lethal process. Diseases like multiple sclerosis, where your immune system attacks your brain, or attacks many organs in your body, like systemic lupus. So there are a number of diseases like that that are important. But it also sets up something else, because normally when you're infected with a virus, you'll have an inflammatory response. That inflammatory response resolves, and then the tissue repairs, and the immune system helps repair those tissues as well. So the physical signs of an inflammatory process include edema, that's the swelling of the tissue, erythemic, it gets red. Anybody ever have inflammation? You'll know it's painful, all right? If you do a heavy workout, you'll get tissue inflammation in your muscles. It's sore the next day, and maybe 2 days later, depending on how hard you work out. But the other feature in a tissue that's undergoing inflammation is that there's an infiltration of leukocytes, of white blood cells. Normally, the white blood cells stay associated with the blood vascular system unless they're called for some reason, a tissue damage, a viral infection or something. They leave the blood vascular system and into the tissue. And from there, they move back in through the lymphatic tissue, through the lymph glands, and process the antigen in lymph nodes and other types of lymphoid organs. So a great example of an inflammatory response that you can see in the skin is shown here. This is in an individual that has herpes simplex infection. Now, herpes viruses are viruses that once acquired stay with you for your entire life. They have evolved multiple mechanisms of evading and tricking the immune system so they can stay with you. And occasionally they reemerge and cause an infection of which now that can be disseminated to another individual that is naive to herpes simplex, has not been infected with that particular virus. But your immune system is called into action, and it initiates the inflammatory process. So if you look at that on the skin, you'll see it's swollen, it's red, and in the center of it you can see little blisters, and that's where the tissue destruction is occurring. The immune system is attacking the cells that are replicating the virus. And those cells are presenting antigens to the immune system, the foreignness of that virus. And that's what the immune system is responding to. And it's painful. So the organs that are responsible for the immune system are called your lymphoid organs. And we categorize them into two different categories. Those that are primary lymphoid organs, that's your thymus gland. And that's where precursors to your white blood cells differentiate into what are known as thymic-derived lymphocytes, or T cells. And then your bone marrow is another site at which the hematopoietic system, including a group of white blood cells called B lymphocytes, because they come out of the bone marrow. Immunologists are not terribly creative when they come to naming things, all right? They try and keep it pretty simple. So during development, through your first 10 or 12 years of life, if you're a human, this is the two organs that allow the white blood cells to differentiate. And particularly important in this process is that in the thymus, T cells learn to distinguish between what is self, and when they encounter self proteins, those lymphocytes are eliminated from the body. So what's remaining are T cells that recognize everything except self. Now, with that said, I'll tell you an exception to it, is that it's not a perfect system. And most of us in this room will have T cells circulating in our blood system that can recognize self proteins. But they're being kept at control so that they don't attack our own tissues. So it's not a perfect system in that regard. But it shouldn't be, because we're in a constant battle with the environment, evolving, because the pathogens are evolving, and we're not quite as fast as pathogens are at evolving our genomes. Right? So we have to have other mechanisms that will control this process. Secondary lymphoid organs are where immune responses start. Lymph nodes, tonsils, and adenoids. How many in here have had their tonsils taken out? Okay, almost everybody over 40 except for a few. Right? It's not often done anymore, but when I was a kid, if you had swollen tonsils and you couldn't talk, you couldn't swallow, they'd take you to the surgeon, they'd snip them out. You don't really need them to live. And in fact, because they capture pathogens, they're often chronically inflamed. And it can be a disturbing situation if you're a young kid and you've got a sore throat all the time. There's no doubt about it. But with modern antibiotics and treatments like that, and vaccines, it pretty much has eliminated the need to remove tonsils, unless they get so big that they block your breathing capability or swallowing capabilities. In your lungs, you have the bronchus-associated lymphoid tissue, and in your intestines, you have what's known as Peyer's patches. So when you think about the blood vascular system, as the blood pumps through your body, there's always leakage of fluid out of the capillary beds into the tissues. And so there's a parallel freeway system, if you will, called the lymphatics that drain that fluid back into the blood vascular system. At the thoracic duct is where it re-enters into the blood vascular system. But along that freeway system, there are these nodes, and these are collections of where white blood cells take up residence, but they're continually circulating. So if you tap that thoracic duct, you can drain all the white blood cells out of an individual. Right? So it's a constantly circulating system. The average resident time in a given lymph node for a lymphocyte in the model systems that we study and can be very quantitative about in mice is about 8 hours. And the concentration of lymphocytes in this region in a lymph node is incredibly high. It's 100 million cells per cubic centimeter. It's an incredibly concentrated. And these cells are in close contact with each other. And when an antigen penetrates the system, one or more of those can grab a hold of that antigen and begin to undergo an immune response. So these are the organ systems. We have the Peyer's patches that line the intestinal tract. And so the lungs, which are internal, are constantly breathing in environmental pathogens, if you will. And your intestine, every time you go to McDonald's and eat a hamburger, you're taking in potential hazardous substances. Not that McDonald's is hazardous food. It's the microorganisms that you drink and breathe and come into your system. If you think about that in your intestinal tract, it's loaded with bacteria. Yet it's inside you, and you're not having an inflammatory response. And so there are these mechanisms that keep the microorganisms in the intestinal tract without gaining access to the body. Occasionally those systems fail, and you develop an inflammatory bowel disease, Crohn's disease, which can be an autoimmune-like disease as well in that setting. All the orifices in your body have immunologic cells right there ready to respond in certain, but they're continually moving. So the cells of the immune system, as I've alluded to, There are cells of the myeloid lineage. These are part of the white blood cells. Macrophages or monocytes, eosinophils, basophils, and neutrophils. Neutrophils are very prevalent in human circulation. Those are the first cells typically on site of an inflammation. So if you cut your skin, within minutes, platelets are aggregating, setting up a clotting mechanism that helps isolate any microbes that might fall into there. And within 12 to 24 hours, neutrophils crawl out of the blood vascular system into that wound and begin phagocytizing any bacterial particles that they can recognize and take in. Those bacterial particles and the debris from the leukocytes drain into the lymphatics and bring the antigens with them. And it's there that macrophages and monocytes begin to sense that there's something ongoing in that setting. Eosinophils and basophils, let me get a quick hand. How many have allergies? Yeah, okay. Probably more than statistically significant. Eosinophils and basophils carry out the allergic response, and they're typically associated with your lung tissue and give you a very rapid response. Basophils can also be in the skin and can give you a very, very rapid response to an allergen. But you also have adaptive defenses. So I recall that I told you the boy in the bubble had severe combined immunodeficiency. He lacks both T and B cells, those cells that come out of the thymus and those cells that come out of the bone marrow, the T and B cells. These cells have an incredibly powerful system to recognize antigens. The ability to activate these cells goes through several stages. The first stage is antigen recognition. So they have on their surface proteins, we call them antigen receptors, a very defined type encoded in your genome, that can recognize foreign substances, foreign proteins, foreign nucleic acids, and the mixture of those, phospholipids as well. Those receptors drive a signal inside those cells to become activated. That means they become metabolically active. They gear up to undergo cell division. That first period of time for them to kick into gear, if you will, and undergo division takes about 3 days. And then they'll divide again. And they'll divide again. And they can divide as fast as 8 hours. It's the fastest known cell division that we know in a mammalian system, is the ability of a lymphocyte to divide and multiply. And so what happens is that those cells that recognize that particular antigen as being foreign undergo clonal expansion. So an individual cell expands itself millionfold, if not more, 10 millionfold. So it accumulates a large number of those identical cells that recognize that particular antigen. And that begins to drive the immune response. But as they proliferate, they also begin to differentiate. And in the case of a viral infection, some of those cells differentiate into cells that have a killer capability. They have cytolytic function. That is, if they see a cell that is harboring a virus that they can recognize, they kill that cell. And they can kill it very quickly. And that helps eliminate that virus, that source of virus production, if you will, in the host. But they also do something else. A few of those cells will go on to become memory cells. And so the next time they encounter that particular pathogen, they respond much quicker. Instead of taking 3 days to divide, they can divide within 12 hours and expand themselves again. They also take up residence at the site of where they saw that original pathogen. So your immune defenses become educated in where they might encounter that particular pathogen. B lymphocytes, B cells, produce an antigen receptor that they secrete, and it ends up in the circulation and can bind to an antigen wherever it comes into contact with, through the blood vascular system, in your gut. There are multiple forms of these receptors that literally bathe all of your tissues in a protective receptor in that regard. So one of the key aspects about this immune system that we have in terms of antigen recognition is that a scientist, Karl Landsteiner, who won a Nobel Prize for identifying blood groups, type A, B, and O, blood groups. But he also did another incredibly interesting experiment that proved that the immune system is a very creative sense, if you will. What he did is he synthesized antigens that have never existed in the universe before. He used chemical compounds to conjugate to proteins, and he demonstrated that the immune system has the capacity to respond to those chemical compounds that have never existed in our universe. Too bad the Martians didn't know about that. Okay? And maybe we ought to keep that as our secret when we move out into our new destiny. The intriguing aspect about this capacity to create molecules that recognize things that have never existed in the universe is contained in a few genes. So for example, here the antibody molecule has two chains, a heavy chain and a light chain. They're both macromolecules, they're both proteins, and they form a combined element, if you will, that physically binds to a molecular shape. The heavy chain, there are approximately 65 genes that encode what are known as the variable region. There's also a smaller segment in the genome right close by. It's called the diversity region. There's 27 of those in humans. And a joining region, there's 6 genes for that. So that heavy chain, there are basically 10 to the 4th recombinant ways you can recombine those genes to give you 10 to the 4th different types of antibodies. When you include the light chains into that, all of a sudden it takes on an entirely different paradigm. With a few more genes, the combinatorial addition, if you will, moves up to 100 million different types of antibodies your immune system can encode for with a very limited number of genes. With literally just a handful of genes, it can create more than 100 million different combinatorial arrangements of that antibody protein. You can cover almost every structure known in our universe and outside the universe. But there's also another very intriguing feature about the immune system. It has the capacity to undergo somatic mutation. So there's another set of genes, a set of enzymes that will introduce single point mutations into that V region, and it can continue to generate diversity in that antibody population. Because if you think of a virus, it can change very rapidly. And so a single clone that's expanded, if it's stuck in that point and can't change its antibody to continue to combine with that viral protein, you're on a losing side of that war. And so the B cells and T cells have this capacity to undergo somatic mutation. That's a powerful tool for the immune system, but also think about it from a cancer biology point of view. You've got a system in your body to mutate your genes. That's a dangerous proposition. So we have lots of enzymes that try and scan our genomes on a continual basis to make sure we're not mutating. But in B lymphocytes, they're allowed to do this, and T cells are allowed to do this somatic mutation. There's a virus known as Epstein-Barr virus. It's a herpes virus. It infects B cells in humans. And when you combine that virus with the capacity of B cells to undergo mutation, in a small fraction of our population, about 1% to 2%, those individuals go on to develop lymphoma, a cancer of the white blood cells, a cancer of the B cells. So that system can get messed up by a viral pathogen. And that's one of the causes of human cancer, is that system, that combination of systems. So let's go on and talk a little bit more about how we see the immune system and disease processes. So I kind of like this little Venn diagram. sort of the central vortex of human disease, if you will. And right at the center is immunity. Almost all diseases, except for some genetically defined diseases, involve the immune response in some capacity. In some cases, it causes the disease. In other cases, it's a passenger in the process. But nonetheless, understanding those mechanisms of how the immune system is involved in disease gives us an opportunity as scientists and physicians to change the course of that disease. That's really what we're trying to accomplish here in that setting. So immunity we naturally think of as being essential for protection against infection from environmental agents as well as those microorganisms in our gut. But it's also very important for our immune system is involved in sensing whether or not we have mutations in our own genomes. So if you have a liver cell that has been exposed to a little bit too much toluene or too much alcohol over a long period of time, that DNA is going to get mutated. And the proteins that that liver cell produces will show up as a mutation. That's a foreign antigen to that individual's immune response. If the conditions are right, the immune system will recognize that and kill that hepatocyte and get rid of it. Mutation over, it's gone. All the rest of the cells in the body there are relatively normal. If you go over 60, 70, 80 years of life, it becomes a much greater proposition for the immune system to begin controlling all these cells that are being mutated. Because look, we are. There's no doubt about it. You have a limited period of time when cells aren't mutating, and that's usually when you're under 12. After that point, after reproductive capability, it's kind of over. You start aging. And I can say that. You know, I'm 65, so. And I know that. I'm collecting barnacles all the time from being sun exposed and living with that. It's not that my immune system is shot, it's that there are lots of mutations that the immune system can't do anything about. When your immune system is too active, or it loses the controls that keep it from attacking your own tissue, that's what we call autoimmune disease. And cancer is that reflection I was telling you about, the immune system being involved in recognizing cancer. And I'll come back and give you some really, I think, very exciting work being done in that particular area. Now, one of the ways that we approach understanding how the immune system works is that we study how viruses engage the immune system. And so my lab over the last 25 years has been involved in studying how herpes viruses, so you can see now why I keep talking about herpes virus. I know a little bit about those viruses. They are incredible masters of the immune response. And part of their strategy is to tangle with the immune system over evolution and now engage it in a way that maintains its homeostasis. So most herpes viruses cause very benign disease in most of the human population, unless you have some type of immune variation or immune deficiency. Then they'll be lethal. So it's a tango between the immune system and the virus to maintain that aspect. But the genes in herpes viruses turn out to be about half the genome is dedicated towards evading the immune system. And so we've been systematically studying these over many years, and it's given us a lot of clues in how the immune system actually works in that setting from a number of point of view. One of the systems that it's told us about are what are known as checkpoint regulators. And this is, you may have heard about these in the popular press, and let me tell you a little bit about them. So shown over on the far right is a crystal structure of a complex of a checkpoint regulator known as BTLA, or B and T lymphocyte attenuator. That means, as the name implies, it's a molecule that limits the immune response. In the bottom, a long molecule here known as HVEM, that's the herpes virus entry mediator, discovered by a colleague of mine back at Northwestern University. It was discovered because it allows herpes simplex to attach to a cell and then get inside the cell. But it's one of about 7 or 8 different ways herpes viruses get into cells. But this protein turns out to be incredibly interesting because it turns on this inhibitory signal. So the virus, why would the virus want to attach to a protein that turns on an inhibitory signal? Well, it's obvious. It attenuates the immune response to the virus. So it's already learned how to do it. It teaches us about how the immune system works. So that was a really striking breakthrough for us in our laboratory to understand how this molecule works. So turns out there's at least 40 of these checkpoint regulators that the mammalian immune system uses to keep itself in balance so that it doesn't become too active, but it's active enough to take care of most pathogens. It helps us maintain immune homeostasis, or balancing the immune system, if you will. Now, we've been clever enough to design drugs that can block the interaction of these two proteins and proteins like them. And when you do that, the immune system can be activated. And that's an important feature as we talk about cancer, and I'll come back to that. But let me show you a little bit of experimental evidence. So we've been able to create mice that are missing just the BTLA gene. So we've knocked it out. And in this particular model, we're comparing the wild type mice on top to the same strain, genetically identical, except it's just missing the BTLA gene. So it's missing that specific checkpoint regulator. If we shave the hair off of the back of these mice and paint them with a little drug known as imiquimod, of which some of you in here, if you've ever had a wart that you wanted removed or a basal cell carcinoma, that's the drug you use. If you paint a little bit of that on the back, in a normal mouse, they get very little inflammation. But if you're missing BTLA and other checkpoint regulators, they get a very erythemic, swollen back, as you can see in that lower panel. Those are 3 identical mice. That's the experimental evidence. In another setting, now instead of on the skin, we look at the intestinal tract. Instead of knocking out BTLA, we knock out HVEM, just its other partner, if you will, just the gene for that protein. And the panel on the right shows you that the colons of those animals get huge because they have an inflammatory-like bowel disease compared to wild type. And they develop it much more rapidly in the absence of HVEM. Again, it kind of proves this is the same phenotype. It's an enhanced inflammatory response without these 2 gene products. And that's a pretty good piece of genetic evidence that they are a ligand receptor pair and they interact with each other to limit the response of the immune system. And so, as I indicated, there are at least 40 of the other checkpoint regulators. The way they actually work on a cell is they recruit an enzyme known as a phosphatase. And the phosphatase dephosphorylates the receptors that are being activated with antigen. Those of you that enjoy biochemistry and cell signaling work, you'll probably know what I'm talking about. If you don't worry about it. It won't be on any test. That has led to a new type of cancer therapy, one that's had huge impact for patients over the last probably 5 years. So remember, antibodies can be directed at many different types of structures, and we're good enough to make antibodies in rabbits and mice and even in humans that we can engineer into a molecular form that block the interaction between the regulatory checkpoint and its ligand. So they can't get together. The antibody sterically hinders that process. So the immune system loses a checkpoint regulator. We can't do it genetically, we can do it pharmacologically. And what happens is that in people with cancer, their immune system attacks the cancer and gets rid of it. So the electron micrograph here shows a tumor cell of which an antibody to a checkpoint regulator known as PD-1 has been treated with that antibody, and these cells now attack this cancer cell and kill it. So the little smaller cells in red there are the lymphocytes, a specific type of lymphocyte known as a CD8 positive T cell. So I told you about T cells and B cells. Within the T cell and B cell compartment, there are about 20 different categories, subcategories, because these cells differentiate into very specialized functions depending on where they're at in the body and how and what kind of effector functions they're going to have. 2 years ago, or 18 months ago, the Food and Drug Administration approved the use of this anti-PD-1 or anti-CTLA-4 antibody in patients that have malignant melanoma and non-small cell lung cancer. And these antibodies, in about 30 to 40% of the patients, show a remarkable response. Metastasis go away, primary tumors shrink. In about 25% of the patients, you'll see a cure. The cancer is gone. Right? That means 75% of the patients don't have a full response. Why? We don't know. That's a real question that scientists all over the world are trying to address. Trying to figure out what it is, why won't this antibody work wonderfully in everybody. We're not genetically identical. We're very different in that regard. And there are different control mechanisms because the cancers themselves are different. So if we all had melanoma in here, I could tell you right now that everyone's melanoma would be different from everybody else's. Melanoma turns out to have the highest mutation rate of most cancers, on the order of at least 200 to 1,000 different mutations in the genome in a melanoma. Okay? Other cancers may only have 1 or 2 mutations. But some of those mutations occur in molecules that aren't ever displayed to the immune system. Now, having some background in understanding the immune system and this balance that these checkpoint regulators play, what do you think is the side effect for this type of immunotherapy, where you block a checkpoint regulator? So, in fact, what happens is that patients treated with a checkpoint regulator known as anti-CTLA-4 or anti-PD-1 develop autoimmune disease. Psoriasis in their skin, Crohn's-like disease in their intestine, diabetes in their pancreas, thyroiditis in their thyroid glands, other types of endocrinopathy. Basically, almost all types of organs show it. These patients never had any evidence of autoimmune disease. And so from my perspective as an immunologist, we're kind of walking around with the potential for autoimmune disease in all of us, but we don't. It's because these checkpoint regulators know when to turn things off. Again, individuals that do develop an autoimmune disease represent on the order of about 1 to 2% of the population. And so genetics plays a very big role, but so do environmental processes that may accumulate over the lifetime of that individual. The one thing you have to think about with these types of antibody approaches is the patient gets injected with it. The antibody goes everywhere in that patient. Okay? So the entire immune system now really has lost that checkpoint regulation. So we see this inflammatory component. It's a good thing if the lymphocytes are around the tumor cell. And taking off that checkpoint regulator, that blockade, negates the effect of the regulatory T cells in that setting. There are other more direct approaches using different types of antibodies that are in clinical trials now, or at least in experimental systems in which you're trying to turn off regulatory T cells without turning off the effector functions of CD8 T cells in that setting. The point I want to make, though, is that autoimmunity it's not really a side effect, it is a consequence of activating the immune system. And these are in adults that normally have never had autoimmune disease sequelae or shown any clinical signs of autoimmune disease. So one of the things that we're doing at Sanford Burnham Prebys, we've discovered how to turn on these checkpoint regulators. So one of the issues with anti-PD-1 or anti-CTLA-4 is that no one knows how to turn them on. So one of the things that we've been successful with is using antibodies to turn on these checkpoint regulators and dial down inflammation in autoimmune disease. And in preclinical studies in mouse models, in a MS-like model, in a Crohn's-like model disease, in a skin-like inflammatory process. We can use an antibody to specific epitopes on BTLA that down-modulate the immune response in that setting. So that attracted the attention of some major pharmaceutical companies, and we were successful in developing a collaborative environment with, in town here with Eli Lilly, and we're developing one of these monoclonal antibodies to go into lupus and also into MS. Those are the clinical indications for that. So it spurred us to continue our research in this particular area. One of the things, if I can backtrack for a minute and tell you a little bit about the research effort and how long this takes. The anti-CTLA-4 antibody that was approved in melanoma, Jim Allison was a colleague of mine when I was a graduate student and a postdoc in the late 1970s, early 1980s. He discovered CTLA about the same time we were discovering some of these checkpoint regulators in 1993 and 1994. And it took almost 10 years to develop an antibody that he could get into a clinical trial. And in that first clinical trial that they ran, there were about 300 patients. And they compared them with standard of care therapy. And there was about an 8-week survival advantage for those patients over the first 6 to 9 months. But he continued to follow those patients over the next several years. And there was a group of patients representing about 12% of the patients that had received that treatment. This treatment had stopped. They went on to become long-term survivors. And that was the first recognition that they had had an impact on the immune system. But it took another 10 years in order to get that drug approved for therapy. Now it's approved for melanoma. It's also approved for, as I mentioned, lung cancer, breast cancer. Within probably the next 2 to 3 months, the FDA will issue will issue a license to sell that particular drug to patients with triple-negative breast cancer. And I can speak personally about this situation. A very close friend of mine had triple-negative breast cancer, and about 6 months ago, she was literally on her deathbed. She had brain metastasis, she had metastasis in several of her other organs, her lung, and the primary tumor had grown to about 20 centimeters in size. She started on anti-PD-1 therapy. I got a text from her yesterday. Her brain metastases are gone, lung metastases are gone. Her primary tumor has shrunk from 15 centimeters to 5 centimeters, and she's loving life again. So it can have a major impact. I think that's why I'm so excited about this particular type of immune therapy. Again, it goes after regulating the immune system, not the cancer cell directly. So there's no harsh chemotherapy, but there is that consequence of autoimmune disease. So if you're a patient, you got a lethal cancer, a little inflammation in the gut or skin, which one are you going to choose? It's not a question. But in some cases, the autoimmune phenotype actually becomes serious enough where the intestines fuse with the skin, you get these fistulating colon, it's horrible, and you have to stop therapy. So there is still much work that needs to be done to refine these therapies in ways that don't lead to that kind of autoimmune disease in that setting. And because of these personal experiences, you know, it's important to recognize that it's not just the scientists in the laboratory. You have to think about the patients themselves. They're the ones that are the most courageous because they have to get involved in a clinical trial. Right? And it really takes a whole community, the physician, the scientist, the patient advocates, the funding mechanisms, the fundraisers that we do to help support research, help support the patients as they go through these clinical trials. It's absolutely crucial. And so I'll just sort of end on one note. This is what I like to do. Every year I raise money for the Moores Cancer Center, and we have a surf team, and last year we won the contest. I beat Rob Machado's team. So, and the people that really do the work are shown here. This is my laboratory. at the Sanford Burnham Prevost. And if you ever have questions, you can always email me, and I'll hopefully get back to you. So thank you very much for your attention.